Welcome back everyone, Miss Portelli here. Today's video is going to be focused on our blood glucose regulation mechanism. So we're still looking at the dot point that we've been focusing on for quite a while now, finishing off by looking at how our blood glucose levels are regulated. Before we do move into the stimulus response models that we will need to focus on for our blood glucose regulation, there's just a couple of things that I wanted to highlight to each of you to make sure that we are starting this off on the same page. First of all, I wanted to clarify that when we are referring to blood glucose regulation, our blood glucose levels are the amount of glucose that we can find in our actual bloodstream. Okay, so we're looking at the individual glucose molecules that we're going to find in our bloodstream. So you can see these two little pictures at the top here sort of pointing to some blood vessels that have little molecules in them. Those little molecules are representing our glucose, or the glucose concentration at the time of the image being taken. The second thing that I wanted to point out to you is obviously these blood glucose levels are going to fluctuate in humans, or any animal really. And the reason that they are going to fluctuate is our end goal is always to get those glucose molecules out of our bloodstream and into our cells so that our cells can use them for different metabolic processes. In particular, our cells will use glucose in the process of cellular respiration so that it can generate energy, or ATP, for that cell to survive. The last thing that I wanted to point out on this slide here is we are going to be referring to two hormones during the rest of this video, and those two hormones are glucagon and insulin. I'm fairly certain most of you would have heard about insulin before. We talk about that heaps in terms of blood glucose regulation and talking about diabetics, which we'll refer to in another video. The less known about hormone would be glucagon. Both are still really, really important, and both are involved in maintaining our blood glucose levels. Also, both of these hormones are produced by and released by our pancreas. So we discussed our pancreas when we looked at the digestive system. Obviously, it was responsible for releasing lots of different digestive enzymes. Well, another function that it has is that it produces these two very important hormones that we will eventually use in maintaining and regulating our blood glucose levels. So here we have just a little bit more information about these two important hormones and what their overall role is in blood glucose regulation. The first one, insulin. This one is super important because this is the hormone that actually allows our cells to absorb that glucose from our bloodstream. Okay, so whenever our cells want access to that glucose, insulin needs to be involved to allow those cells to access it. And if you look at the diagram just underneath that heading there, we can see that these insulin molecules here have been produced by the pancreas. Those insulin molecules will float around our bloodstream until they are needed, and they will actually interact with receptors on the surface of pretty much every single body cell in our bodies. And the reason they'll do that is because every single body cell in our body needs access to glucose, because they all need glucose to make ATP, or energy, to keep us alive. So we can see here that little purple triangle is interacting with the pink receptor there, and by doing so, that little purple triangle, that insulin molecule, is actually triggering a protein channel over here to open up and allow that glucose from the bloodstream to enter through the protein channel into the cell. By doing so, that's obviously going to allow the insulin to reduce the blood glucose levels, because it is removing the glucose from our bloodstream. On the other hand, we've got the hormone glucagon, okay, also released by our pancreas, but this time the glucagon hormone doesn't interact with every single body cell. It actually only targets cells in your liver. And it targets cells in your liver because your liver cells have complementary receptors on their surface that, when they interact with the glucagon hormone, will actually trigger those liver cells to release any stored glucose that they might have. Now, we refer to stored glucose as glycogen. I know it does sound very, very similar to glucagon, but they are two different things. Okay, so glucagon is our hormone. Glycogen is just a big chain of glucose molecules stored together in the liver. So if we look at the diagram underneath, we can see that a little bit clearer. We've got our glucagon molecules here, or our glucagon hormones that have been released by our pancreas that are interacting with the liver, or about to interact with the receptors on the liver. And in doing so, they're going to trigger the glycogen molecules, our stored glucose. So we can see that nice greeny blue chain there of glycogen. It's going to trigger that glycogen molecule to break down into individual glucose molecules, just at the bottom of the liver there. And eventually those individual glucose molecules will be released back into our bloodstream, which will overall cause an increase in our blood glucose levels. So insulin is responsible for reducing our blood glucose levels and bringing that glucose into our cells. Glucagon does the opposite. It is responsible for triggering our liver to release stored glucose in the form of glycogen back into the bloodstream, which will have an overall increase in our blood glucose levels. Let's have a look at what happens to our blood glucose levels when our body detects them as being too high. So obviously our stimulus in this scenario is going to be having high blood glucose levels within our body, and that would be detected by receptors that we find in our pancreas. Now these receptors are located specifically on cells called beta cells. Not something that we've talked about before, but it's good to know that term because you'll probably come across it quite a bit if you're doing external reading in your textbook or looking at online resources. So these cells in your pancreas, they have receptors on them that are detecting that change in your blood glucose levels. Now next in our stimulus response model is usually our control center. With this particular example though, it's a little bit different to the other ones that we've looked at, in that our control center is no longer the brain of the organism. We're actually going to refer to it as the brain of the cell. So if we think about what we know about the cell, the brain of the cell or the control center of our cell would be our nucleus. So when our beta cells in the pancreas, or when the pancreas cells have detected that the increased blood glucose levels have occurred, that signal actually gets passed through the cell to the nucleus, and the nucleus will cause the cell to change in some way in response to that stimulus. At year 11, we don't look specifically at how that message gets from the outside of the cell to the nucleus, but in year 12, when you look at cell signaling, you go into that in quite a bit of detail. So I'm just going to leave it there for now. So we've got our stimulus of high blood glucose levels. Our receptors are cells within our pancreas. The beta cells within our pancreas have detected our blood glucose levels are too high. That message has been received by the control center or the nucleus of the cells. And then our effector, those cells of the pancreas again, start secreting insulin, or they start producing insulin. Remember, insulin is that hormone that allows our cells to be able to absorb the glucose from our bloodstream. So the beta cells within the pancreas produce that insulin. That insulin is then going to interact with all the other cells throughout our body and give those cells the ability to now absorb the glucose from our bloodstream, reducing the amount of glucose that you find in the bloodstream. So we end up with an overall decrease in the amount of glucose in our blood, which is the opposite to what we started with. Okay, so this is actually an example of a negative feedback loop. Okay. Now let's have a look at what happens within our body when our blood glucose levels are too low. So again, our stimulus is going to be blood glucose levels being low, or a decrease in our blood glucose levels. Our receptors are still going to be receptors in our pancreas, This time, it's focusing on a different type of cell. So in the previous example, when our blood glucose levels were too high, we were looking at beta cells in the pancreas, so a specific cell type in the pancreas that detects when it's too high. This time it's a different cell type. It's alpha cells in the pancreas that are detecting when it's too low. Still doing the same thing, they're going to receive that signal as the receptor, and the control center, just like we talked about in the previous example, is still going to be the nucleus of those cells in the pancreas. Okay, so we're not sending it to the brain of the organism, we're sending that signal to the control center or the brain of the cell, which is our nucleus. Once the nucleus has received that message, it's then going to trigger our effector, and in this case it's those special cells within the pancreas, the alpha cells, and they are going to secrete or produce a hormone, and that hormone in this example is glucagon. Okay, so remember, glucagon was the hormone that interacts, so once it's produced by the pancreas, that hormone glucagon is going to go and interact with the liver and cause the liver to break down and release those stored glucose molecules. So I guess you can probably guess what our response is. The glucagon is now causing the liver cells to break down the stored glucose, which we referred to as glycogen. And that stored glucose gets released back into our bloodstream, which would give us an overall increase in our blood glucose levels. Okay, so our response has resulted in increasing our blood glucose levels when our original stimulus was a decrease in the blood glucose levels. So we've brought about a response that's opposite, or reducing or stopping the original stimulus. Okay, so again, still looking at an example of negative feedback.